Geneticists are very interested in mutations. Now, as we'll see in this video, there's lots of causes of mutations, lots of effects of mutations, but let me just start off with this one. Say if in an individual there was a change in the DNA sequence on, say, a gene called P53 located on chromosome 17, this change in the DNA sequence would then result in a change in the sequence of the mRNA, which was transcribed from that DNA. And this could then lead to a change in the amino acid sequence in the protein. And if that protein works differently or is non-functional, then that could result in cancer and change phenotype. So mutations are certainly interesting in as we search for the causes of disease. But then also, as we just consider life on Earth, I mean, mutation is a major source of variation in populations and ultimately the origin of new species. And since we humans are mutated apes, and apes are mutated catarrhine primates, and mammals are mutated reptiles, certainly at some point then mutation is a major source of the variation of life on Earth, the diversity of species which we have on Earth, etc. So what are then the causes of mutations? Well, there are a lot. So I'm just going to run through a whole series of ways that mutations can be caused in no particular order. So they can be caused by environmental substances. And so there are what are called mutagens. So there's a molecule called 5-bromouracil, which looks enough like thymine that a replicating cell could insert this molecule instead of thymine in an elongating DNA chain. But the difference is, while thymine binds to adenine, bromouracil would bind to guanine, and this then would lead to a change in the DNA sequence of the daughter molecule. And there are plenty of other examples of this. There are what are known as hydroxylating agents, which can add OHs to a group. If an OH is added to cytosine, that will change its pairing. So this is a modified cytosine. The original cytosine would have paired with guanine. This modified cytosine will pair with adenine. There are mutagens which can add either ethyl or methyl groups, and as a result, an altered thymine could bind to guanine. An altered guanine could pair with thymine. Amino groups can be removed, so cytosine could bind to uracil. Adenine could bind to hypoxanthine, which will later pair with cytosine. There are mutagens in the environment which can cause bases to be added or lost. And so one of the causes of mutation is things in the environment that we can be exposed to. Now, just very quickly, I'm trying to focus on mutations. Obviously, such a broad topic could then, take us into different avenues. Where might you be exposed to these mutagens? Well, you might say, oh, well, perhaps I would be exposed if I was, living near a chemical, waste plant, or, eating fish from a river where industrial waste had been released. Those are both true. But also, you can just encounter them in typical daily life. So, for example, if you eat plants, plants, and so this is a separate conversation, but I always tell my students, sorry if this affects how you view the world. Plants don't necessarily like you. I mean, you're an animal. You eat them, and they would like you to stop. And if you're a plant, how do you get an animal to stop eating you? You can't swat it away. You can't move. But what you can do is poison your leaves. You can put nasty things in your leaves so that if an animal were to eat a large quantity of that plant, that there would be a negative consequence. So as a result, a lot of things taste bitter to us because your tongue is warning you, don't eat this plant. It's trying to poison you. That's what bitter taste receptors allow us to perceive. So if you were to look at broccoli or coffee, they can have a bitter taste. There are mutagens there, low levels, all right, because these have been cultivated, not like wild plants. So if you were to just walk through a field and start putting wild plants in your mouth, they would taste very bitter. There would be far more toxins there. They haven't been, cultivated through agriculture. But we could find then, even in, the plants that we get at the supermarket, low levels of mutagens there. When we, cook meat, the charred ends of meat can have mutagens there. And so this is something which affects us, even if you're not living next door to, a chemical, uh, you know, manufacturing plant or, a Superfund site, et cetera. So we do encounter things in our environments, and wild animals do as well. If they're eating plants or eating the animals that ate plants, they're encountering these mutagens. Another cause of mutations. Now, this is an odd one, so we'll zip through it. And if you study chemistry, this is always a lot of students just, this is a bridge too far for many of them. You study these chemical molecules and you give them formulas with the idea that this is where the hydrogen is and this is where the electron is. And you have this idea that they stay. And the truth is they just don't. If you have a big molecule like this, electrons can move around it. That these molecules will sometimes, change, their conformation, lose a hydrogen, gain a hydrogen. And so an amino group, which is NH2, can change to an amino group, which has a double bond and is NH. It happens. It's just a spontaneous base change. The molecules which we draw on the paper, they don't necessarily stay in that conformation. There are major and minor forms. There are tautomers of molecules. So when we draw a molecular formula, so this molecule usually looks like this, but sometimes this hydrogen gets lost, this gets added, it just happens. Electrons move. These molecules are not as stable as we think they are. And if then that adenine underwent that spontaneous change, it now binds to cytosine rather than thymine. So these spontaneous changes, which occur naturally, they're not induced by anything. These are just minor forms of adenine, minor forms of cytosine. They bind to different um, complementary nucleotides, and this would then cause a change in the genetic code, a mutation. So mutations will occur, period, without any other external force, just because these DNA nucleotides are not as stable as we think they are. If, just a reminder, in the DNA nitrogenous bases, there are two that have a double carbon ring here. So this is adenine, this is guanine. They have a double ring, 1, 2 rings there. So they are called purines, while cytosine and thymine and an RNA uracil, they just have one ring. That's what's called a pyrimidine. If a mutation changes a purine for another purine, that's what's called a transition mutation. If a pyrimidine is replaced by a purine or a purine is replaced by a pyrimidine, that is what's called a transversion mutation. And so keto groups can change to enol groups, which are just an OH, and this can then change the pairing here. And so just another source of mutation is just spontaneous nucleotide changes. There are major and minor forms, and as a nucleotide transitions from one form to another, this can result in transition mutations, where if you look, the pyrimidine here, thymine, is replaced by another pyrimidine, or this purine is replaced by another purine. They would be transition mutation, while transversion mutations would change a pyrimidine into a purine. Um, so that's a cause uh, mutation. Another cause of mutation is, I often ask, students, like if you were to take your textbook and try to type it, so you sit at, a computer and you try to uh, type it letter for letter, word for word, you're going to make typos every now and then. And when um, the S phase occurs, your uh, nucleus is essentially trying to copy the DNA code. And so you convert a double-stranded DNA molecule into single-stranded DNA molecules. And the DNA polymerase is now trying to only insert adenine to pair with thymine, and only insert thymine to pair with adenine, so that um, there are these complementary base pairs. Well, if you were to try to type an exact sequence, you make mistakes from time to time, and so does DNA polymerase. It's not perfect. And so therefore, there are cases where then abnormal, the incorrect bases are paired with the original bases. And so DNA polymerase will do this maybe once every 10,000 times. Now that sounds pretty good, but when you take into account that you've got 3 billion nucleotides, if when cells divide, you're getting a mistake every 10,000 nucleotides, and if in that second, you're maybe making upwards towards 10 million new cells per second. That's an incredible number of mutations. This would be very problematic. So luckily, DNA polymerase has a proofreading ability. See, if the pairs, aren't complementary, the shape will be odd. It'll bulge a little bit, and it could be recognized that this doesn't quite fit. And so then DNA polymerase can then proofread itself and then insert the correct nucleotide. So after this, then there is a mistake only per 1 per million um, nucleotides, which is far better. And then there are other proofreading mechanisms, which I'll get to in a subsequent video. And after they act on mistakes which were made, now the only changes to the genetic code occur once every billion nucleotides or more. But once again, if you've got 3 billion nucleotides and you're making a mistake when all of this is done, one per uh, 1 per billion nucleotides, that means every single cell that you make in your body is a mutant cell, all right, because that there was a change in the genetic code. This is why cells which reproduce most quickly, therefore, are at the highest risk of mutation, because even in normal cells, just normal cell division will result in mutations. So in humans, skin cells divide at the basal layer of the epidermis maybe once a day. Skin cancer is the most common cancer in humans. Breast cancer is far more common in women than it is in men, where it is rare. And one of the reasons is that every menstrual cycle, estrogen causes breast cells to divide, so the number of cell divisions is higher in women than in men. So once again, another cause of mutation is that when we replicate our DNA, mistakes are made because DNA polymerase isn't perfect, even after its proofreading mechanism and other error repair mechanisms, which I'll get to. In 1991, there was a new type of mutation which was found in humans. Now, once again, I'm a little older, so this is perhaps a little more significant to me. When I, was going through school, everything was you had to study E. coli first, you had to study fruit flies first, you had to study yeast first, and then maybe what you learned from your study of these simpler organisms could be applied to humans. Maybe you could understand the complexity of humans after learning something in these simple organisms. But by the 90s, in the Human Genome Sequencing project, et cetera, it was now evident that we understood human genetics enough that sometimes we were first discovering things in humans that then we're applying to other animals and other organisms. So once again, perhaps you don't have the appreciation for that, but that was new. That was just a sign of how far genetics had come, that we could now, study the complexity of human genetics and learn things which could be applied to other organisms, where, for, the first century or for almost a century, that was definitely not the case. And what was discovered was that a place which mutates frequently is what's called a trinucleotide repeat area. So, for example, in Fragile X syndrome, there is an area uh, where there's these trinucleotide repeats, CGG, 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 CGG. So the genetic sequence just has this triplet over and over and over again. Normal individuals have 50 repeats in a row. If that amplified to become 50 to 200, you were considered a carrier for the disease. And now your children could then potentially possess hundreds to thousands, which would mean that they would have the Fragile X syndrome, whose consequences would include retardation. And it turns out that that's known in a number of diseases. Huntington's disease can be caused by sequences of CAG over and over again, myotonic dystrophy, CTG, and other examples. And the reason seems to be that if you've got the same kind of sequence over and over and over again, when cells do crossing over in meiosis, that the chromosomes align, but if you've got all of these repeated sequences, they might not align perfectly. And now after crossing over is done, one of the chromosomes now might have um, extra copies of that repeat, and so that it has amplified. That could potentially change the shape of the protein. That could, change a number of things. So once again, this is a type of mutation, that unequal crossing over in an area where you have these trinucleotide repeats will make that change in that gene more likely. And once again, kind of interesting because it was first discovered in humans and applied to other organisms. Now, we could go on and on because there are lots of causes of mutations. I'm just going to jump through them and then get into their effects. And so you could have the sugar phosphate backbone, but you could have the nitrogenous base be spontaneously lost. In depurination, it's the purine base which is lost. Now that just happens, molecules aren't, glued together permanently, so this can happen. And the purines are replaced, but because they're replaced at random, you could then replace an incorrect your substitute could then be the incorrect purine, say adenine instead of the guanine, which was originally there. X-rays can break sugar phosphate backbones and cause a DNA molecule to break, which could lead to chromosomal mutations, which I'll get to later. Ultraviolet light can strike us. And if you have two thymines next to each other, thymines are there in blue, it could cause them to adhere in what's known as a dimer. So they would stick together. And the problem is they were two separate nucleotides, but the next time your cell replicates this DNA, it could perceive these two as one single nucleotide. So exposure to ultraviolet light increases the risk of mutation and cancer. So thus we wear sunblock and try to avoid sunburns and the like. There are those mobile genetic elements. So we like to think of our DNA as stable, but there are these, jumping genes, if you will, a section of DNA which codes for a protein, which will help excise that sequence and insert it somewhere else. So this DNA could have woken up today on chromosome 17, but by tomorrow it could be on chromosome 5. DNA can move, all right? Well, as it moves, it's now changed two sequences. All right, it's inserted over here. What did that do? Did it disrupt a gene? Did it disrupt the regulatory element of a gene? So transposons, these jumping genes, can then make changes. Um, we know that DNA can get transcribed into RNA, but sometimes the mRNA molecules can then be inserted back into the DNA in what's called retrotransposition. So that would disrupt the area where it goes, and it would then also affect gene copy, which I'll get to. Viruses can insert in the genome. So there are endogenous retroviruses, ERVs, that can insert themselves into a host DNA sequence and disrupt genes when they get there. Heat can cause bases to be lost and can cause the loss of amino groups converting cytosine to uracil. We're exposed to reactive forms of oxygen, and reactive forms of oxygen can change base pairing. And so the point is that there are so many different causes um, mutations. Now, I don't want to go back to here because these were topics covered otherwise. I've been focusing on, changes in, DNA sequences, but keep in mind that we also have what are called chromosomal mutations, where an incorrect sorting of chromosomes, what's called a non-disjunction, could lead to too few chromosomes, too many chromosomes, maybe one or two too many or too few. That's what's called a aneuploidy, or maybe a whole extra set. That's what's called polyploidy. There are also changes in chromosome structure. So remember that something like X-rays or viruses or jumping genes, can break DNA when they insert or act on it. Well, this could cause sections of a chromosome to be deleted. It can cause areas to be duplicated. It can cause parts of chromosome, a chromosome, say 5, end up on chromosome 17. That's what's called a translocation. If something breaks, it can reinsert upside down. That's an inversion. So there are sources of chromosomal mutations as well. And if we were to, go into other, organisms, when we look at, say, bacteria, we know that they can, change through things like transformation. They can take DNA up from the environment. There's transduction, where a virus could accidentally insert a DNA sequence from its previous host into a new host, but actually accidentally move a gene from bacteria 1 to bacteria 2. There's conjugation, where bacteria can fuse confuse, and this can insert new genes. So the point being made is that there are a great diversity of potential causes of mutations, of changes in one's genetic code, and so to the point where we are all mutants. All right, so if just from the spontaneous the errors in DNA replication, when your cells divide, as they are right now, each daughter cell potentially has a mutation in it. So, your cells are mutant, and we were conceived with mutations. All right. So the last paper I read on this is well over 100 new mutations in each of us, which were not present in the somatic body cells of our parents. So mutations occur through lots of mechanisms and are so common that you will have mutations occur in your body today, and that you are a mutant compared to your parents. There were changes in you. And so then this obviously gets us to the second topic. Well, if mutations are that common, what what's the effect of these mutations? What do mutations do? And the answer is it depends. So just forgive me, this is the first analogy that just jumped in my head while I'm talking. If you were to ask, like, let's say that there was a disruption of traffic, like a car accident or something in your town, what would that do? Well, that depends. Where did it occur? Is it in an alleyway, in which case minimal effect? Did it happen on a minor street, a major street, a highway? And so, um, the same event which occurs in different places could have very, very different effects. So there's not one effect of mutations. So let me just start talking about things that mutations can do. And once again, I hope you leave with the idea that wow, there's a lot of potential effects of mutations. So recall from our codon chart that in the genetic code, GUU codes for valine, GUC codes for valine, GUA codes for valine, GUG codes for valine. So as a result, if there was a mutation which changed GUC, all right, to GUU, then it would still code for valine. So even though there was a mutation, even though there was a genetic code, a change, changing say GUC to GUU, there would be no change in the amino acid sequence of the protein. And so therefore, it could be a silent mutation, which has no effect. However, if we changed, instead of this second nucleotide, if we changed the first nucleotide, so now say GUC became CUC, that codes for a different amino acid. That codes for leucine, not not valine. So that has changed one amino acid into another. That's what's known as a missense mutation. A missense mutation still codes for an amino acid, but just a different amino acid than the original. If a mutation changed AAG to UAG, that's one of the three stop codons. We call that a nonsense mutation, where a codon which should code for an amino acid would now code for a stop codon. And now a cell trying to make a protein would get to a stop codon prematurely, and it would make what's called a truncated protein, a protein which was shorter. And this protein might not have any function. It might not be able to work. And so, for example, if you were to look at different mutations which can cause breast cancer, some instead of inserting the normal amino acid arginine, then have a stop codon instead, and then the protein is too short, and that it would not function. And so there are lots of examples of that where the mutation is a nonsense mutation which stops the protein prematurely. Here are these other ones where proline is replaced by threonine. That's a missense mutation. So that's an incorrect amino acid which is inserted. Okay. Now, if you were going to insert a different amino acid, what effect would that have? Well, that depends. All right, and so therefore, so once again, I'm sorry, let me just repeat it again. Let's say you were to take proline and change it to arginine, or arginine change it to aspartic acid, or aspartic acid change to threonine. What does that do? Once again, there's no easy answer to that. It depends. So some parts of a protein are more important than others. So for example, enzymes have an active site, a pocket where the reactant has to fit. If there was a mutation which affected the active site, that would have a serious effect on the function of the protein. Now, not necessarily a bad one. So for example, there were digestive enzymes, serine proteases, And a mutation in one once upon a time now made a different serine protease, which ended up being efficient in blood clotting. So it's a different enzyme doing a different reaction. So sometimes, mutations, they don't have to be bad. But once again, there would be a greater effect if we changed uh, the uh, the active site of the enzyme. So once again, there are digestive enzymes, but then these can then be mutated um, and have other functions. And then if we're changing one amino acid for another, the question is, well, how similar is the new amino acid to the old one? So some amino acids are non-polar. And if you change a non-polar amino acid for a different non-polar amino acid, maybe there's no effect or minimal effect. Some non-polar amino acids are just kind of holding the place within the membrane. So as a protein goes through a cell membrane, say if it's a receptor with ends on either side of the membrane, the part in the middle just has to be non-polar because the phospholipid tails of the cell membrane are non-polar. So if we substitute a non-polar amino acid for a different non-polar amino acid, then maybe there's no effect. Or if we change a polar amino acid for a different polar amino acid, once again, maybe there's no effect. In sickle cell anemia, which is a common example of mutations, glutamic acid, which is an acid, is changed into valine, which is non-polar. So that would be a serious change. If you remember about, a protein shape, when proteins, the parts of a protein fold, upon each other, very often it is two cysteines that are then forming a disulfide bond because they have sulfur here. So the sulfur on this cysteine combines with the sulfur on this cysteine, and that's why the protein folds the way that it does. But if you change that it's no longer a cysteine, then the protein isn't going to fold naturally. Sometimes you have what's called a salt bridge between an acidic amino acid like glutamic acid and aspartic acid and a basic amino acid like lysine, arginine, or histidine. So if a mutation changed one of these, and if you were relying on a positively charged amino acid attracting a negatively charged amino acid for the protein to fold the way that it does, then that would change the shape of the protein. So thus the effects um, are different. So that's one of the difficult things for beginning geneticists. And once again, to get back to this video, remember the proteins are big things, right? So P53 or any it's a sequence of amino acids, and a mutation would be potentially a missense would change one amino acid to a different one. So each of those letters corresponds to an amino acid. What are the effects of changing that valine, say in that spot, to an arginine, all right, or to a histidine, or to a leucine? So not only do you have all of these spots, but if they're changed to a different amino acid, there's 20 different amino acids to choose from, all right? And so therefore, there's all of these different possible effects of a mutation. Changing this end of the protein could have a different effect than changing the middle or the other end of the protein. Changing this spot to a serine might have a different effect than changing it to an arginine. And so there's all of these different types of possible effects of mutations. Before I get to my videos talking about how a mutation might have an effect depending on where it is in the gene, let's just go through a whole bunch of definitions once again, just to show you how many different mutations there might be. A point mutation could change a specific spot in a sequence, but then you can have additions and deletions, which can add one nucleotide, but maybe a bunch. Maybe a bunch of nucleotides got added. Maybe a bunch of nucleotides got removed. And this could then cause what's called a frame shift mutation. Because if you remember, those codons, are read every three. So DNA is essentially a language which with three-letter words. And if you have a sequence of letters where like the red dog sat on a mat or something. So if everything's kind of like three letters, if you insert a letter, all right, so instead of the red, you take away the E, now you're reading the TH from the, but then you're putting in the D for the dog. You're like the duh, ugga, and now none of your words make sense. So if you have what's called a frame shift mutation, it changes the reading frame of all of the codons after that. So it doesn't just affect one spot, it affects all of the spots after that. All of them will be read incorrectly. Uh, so, um, um, so that's a type of mutation. If uh, a mutation results in no protein being made, all right, so maybe it's a mutation in the promoter, so the gene is never transcribed or whatever, then that's what's called a null mutation. Sometimes a protein is made, but just less of it. And so that could be a hypomorphic a mutation. Some mutations are lethal, so it could be called a lethal mutation. Some are called conditional lethal mutations. So sometimes, and there are experimental organisms where like the bacteria live, but if you raise the temperature, then they die because it's a temperature sensitive mutation. Or they need certain nutrients. So as long as you give them arginine, they're alive. But without arginine, that they die. Um, and so those are types of mutations. Sometimes you could have a mutant phenotype caused by mutation number one, but a mutation number two could then change it back to the original wild type form, or at least the wild type function. And so that could be what's called a reversion mutation. Sometimes it's in the same gene. So a mutation in the same gene as the first mutation reverts it to the wild type functions. The protein works again. So that could be an intragenic suppression of a mutation. So the second mutation occurs in the same gene as the first, and it gets you back to normal. But maybe a mutation in a second gene then gets you back to wild type function. That would be intergenic suppression. And so there's all of these variations to consider as well. But I would like to, in my playlist, and I know my various nucleotides are speaking with accents just to try to keep my students awake. But this then asks, all right, what would be the effect of different mutations? Well, where in the gene are they? So let's consider a gene. Here's the upstream region. Here's the enhancer area. Here's the promoter. Here's the leader sequence, the translated region, an intron, a translated region, a trailer, the downstream region. Let's say there's a mutation that occurs here in the upstream region. What effect might it have? Well, if it breaks the DNA, obviously that's bad. But otherwise, if you change just one nucleotide for another, because this area never becomes protein, and it's this area which is involved in the regulation, maybe this has no effect at all. So changing a thymine to a guanine might have a big effect here or there, but it doesn't have any effect there at all. The promoter, I'm sorry, the enhancer region often increases the production of a protein product. So this is where transcription factors bind to start transcription or to enhance it, so that more protein is made. So a mutation here could change the amount of protein made. You're still making protein; it has the right sequence. You're just making a different amount of it. So that could result in cancer. You're making too much of a growth factor or too little of a tumor suppressor protein. So now that could then have that effect. The promoter region is where RNA polymerase binds. It's essentially the DNA sequence which says a gene starts here. So you need that tot box, if you recall. And you have so many thymines and adenines because with their double bonds, it's easy to pull the double-stranded molecule apart. But if you were to have a mutation which puts a guanine there, that might mean that you changed this, and it's no longer recognized as a start sequence. And so you just never, the gene is no longer active. And so that could, have a devastating effect, obviously. In between the promoter and the first start codon, the AUG, where you would start translation, is what's known as the leader. All right, so the small subunit of the ribosome binds to the GTP cap of the mRNA, and it slides along to where it finds the first AUG codon, the start codon. Let's say that's here, where the blue begins. Well, this leader never becomes protein. So if you have a mutation there, would it matter? No, not necessarily. Unless maybe if you started a new start codon. So if you just by random luck made that into an AUG, now the ribosome would think that this was where the protein started rather than here. And so especially since you could change the reading frame, this could mean that your protein would be completely different. Okay, so what would be the effects of a mutation here? Well, that depends. So this is the start codon AUG. This is where the first amino acid will be inserted for translation. If you change that, so it's no longer an AUG, then translation won't start there. It will start at the first AUG, which is found randomly later. Once again, the protein could be completely different. If you changed a nucleotides here in the translated region, what would that result in? Well, that depends. Once again, some mutations are silent. So if you take one of the codons for valine and you change it to another codon for valine, you still insert valine. So you haven't really changed the amino acid sequence. But if you make a nonsense mutation, then the protein would stop early and you would never translate this region. If you had a missense mutation, you would insert the wrong amino acid. And so the potential effects here would vary. There are introns which will be removed. All right, so when this gets converted into mRNA, we will only put the blue exons together and remove the introns. So a mutation in an intron, what would that do? Well, maybe nothing, all right? However, that's a little simplistic because the introns sequences are important. Like it might fold a certain way and that helps it be removed. There are branch points and start and stop points. So the very beginning of the intron, the very end of the intron, they are important. If you mutated there, then maybe it's considered still to be an exon and not an intron. And thus, then that whole sequence would be added to your protein. So there's all of these. Now, at the end here, at the end of the translated portion, we will have a stop codon that will tell translation to stop because there's no matching amino acid and tRNA, which can be inserted there. But if you change a stop codon so that it's not a stop codon, translation will keep on going. And now you'll just be sticking all of these additional amino acids in the sequence, which weren't originally supposed to be there, and the protein would be much longer than it was supposed to. In an mRNA, there is a leader before you start translation, and then there's what's called a trailer after the translation should end. But somewhere in here is the sequence that ends transcription. So once you start transcription, at a certain point, you stop. So a mutation in this trailer might have no effect, but it might now make a much longer mRNA because in transcription, you could just keep going. And that would mean that you were less efficient. You could then make mRNAs which were much longer than they otherwise should be. And then once again, I go into in this part the other things like when we consider a protein, some changes are more important than others. So as I said, if this is beta hemoglobin, the valine substituting glutamic acid, which is an acidic amino acid, obviously, for valine, which is non-polar, that could have a serious So valine is nonpolar, while glutamic acid is obviously acidic. So um, the changing one acidic amino acid for another would have a lesser effect potentially than changing an acidic amino acid for a valine. But then even those introns, et cetera, and where transcription ends, RNA can fold upon itself and take on this three-dimensional shape. By changing the shape of the RNA, that might change the splicing of genes or where transcription ends. So to sum up, mutation is a fascinating thing to study. Not only is it important for diagnosing disease, but then understanding diversity of life. All right, so when we look outside, we see different species. Um, we see variations within species. Life changes over time. So ultimately, these mutations are responsible for what we see outside. They're what have made the first mammals different from their reptilian ancestors, the first primates different from their generalized mammal ancestors, the first apes different from catarrhine primates, the first humans different from apes. All right, and so mutation is important, obviously. Then to study, there's not only a broad diversity of sources of mutation, things that can change the genetic code, but then depending on where these mutations occur and what types they are, there's a great number of potential effects.